Glycolysis is the first step in cell respiration, and it's the process of harnessing energy in which one glucose molecule is broken into two pyruvate molecules. We'll say those are pyruvate right there, in the cytoplasm of the cell. So it doesn't happen in the mitochondria, just in the cytoplasm. All the reactions in glycolysis are catalyzed by a specific enzyme. Glycolysis is energy requiring, so it doesn't proceed without the input of any energy. So this energy is made available when two ATP molecules, there's one and there's the other, are hydrolyzed. So the first ATP attaches a phosphate group to the end of the glucose molecule, and it transforms the molecule into G6P, which is glucose 6-phosphate. After this, glucose 6-phosphate gets rearranged into its isomer, boom, fructose 6-phosphate, and the second ATP attaches a phosphate group to the other end of the molecule, right there. So this forms fructose 1,6-bisphosphate. What? And essentially, each ATP at the beginning transfers one phosphate group to the 6-carbon backbone. So the backbone of F16BP splits to form DHAP and G3P. So Tayler is the G3P in this situation, and Allie is DHAP. So then the DHAP gets converted into another G3P molecule pretty much right away. So this results in two formations of G3P molecules. So each G3P gives up one hydrogen and two electrons to NAD, and this reduces NAD plus to form NADH. That's an A. Jeez. So G3P combines with inorganic phosphate, and this results in an unstable molecule, 1,3-BPG. So 1,3-BPG gives up a phosphate group to ADP, forming ATP. This results in a molecule of 3PG. So in the end, two ATP molecules are formed, one for each G3P, and with this formation of two ATP molecules, the original energy investment is paid off. So 3PG rearranges to its structurally different isomer to 2PG. Let's just watch that again. Yeah. 2PG gives up one proton and also a hydroxide ion, which forms water. And the resulting molecule is PEP. Yay! So the molecule PEP is unstable and breaks down, releasing enough energy to transfer a phosphate group to ADP, forming another ATP molecule. So now we've created two ATP molecules for each PEP molecule. At this point, the glucose molecule has been broken down to two pyruvate molecules with 3-carbon backbone. Nice bell.
The second step in cell respiration, pyruvate oxidation. Pyruvate molecules enter the matrix of the mitochondrion with the help of a membrane carrier to pass through the outer and inner mitochondrial membranes. Coenzyme A attaches itself to the acyl group of the pyruvate molecule. This enables it to be oxidized, while NAD is reduced to NADH. The remaining carboxyl group is removed as waste in the form of CO2. resulting in acetyl-CoA. When energy is needed, acetyl-CoA enters the next stage.
The Krebs cycle is the third stage in cell respiration. It's a cyclical cycle, meaning it works in a circle of 8 reactions. It transforms energy from organic molecules into usable forms for the cell. Some of these molecules include ATP, NADH, and FADH2. It also releases CO2 as a waste product. Acetyl-CoA reacts with oxaloacetate, forming citrate, and the CoA is removed. Citrate undergoes isomerization and forms isocitrate. Isocitrate is oxidized to become alpha-ketoglutarate. NAD plus ions are reduced, forming NADH. Alpha-ketoglutarate forms succinyl-CoA by binding with CoA. Alpha-ketoglutarate is oxidized by NAD+, which reduces to NADH. CO2 is released. Succinyl-CoA loses its CoA and becomes succinate. A phosphate displaces CoA, and GDP is phosphorylated to become GTP. A phosphate group is transferred to ADP, creating ATP. FAD oxidizes succinate to form FADH2. Succinate forms fumarate. Water is added to form malate. Malate undergoes a redox reaction with NAD+, producing NADH and oxaloacetate. The cycle repeats. Dun, 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 dun. Krebs cycle is done.
Electron transport chain, otherwise known as ETC. The purpose of ETC is to synthesize ATP through oxidative phosphorylation. Proteins in the ETC are set up in order of increasing electronegativity. Electrons, or energy, are passed from one component to the next along the membrane. The first component is a weak electron acceptor, and the last component is a strong electron acceptor. NADH binds to NADH dehydrogenase. FADH2 binds to succinate dehydrogenase. Hydrogen ions are pumped into the intermembrane space. Mobile electron carrier UQ carries electrons from NADH dehydrogenase to cytochrome BC1, which is the third complex. Mobile electron carrier site C carries electrons from cytochrome BC1 to cytochrome oxidase, which is the fourth complex. FADH2 works the same, but starts at the succinate complex. Since electrons from FADH2 pass through fewer electron carriers, it makes less ATP. Each molecule of NADH produces 3 ATP and 3 hydrogen ions. Each molecule of FADH2 makes 2 ATP and 2 hydrogen ions. When the 2 electrons leave cytochrome oxidase, they can combine with oxygen and hydrogen to form H2O. This water is waste. Oxygen is used because it is the only element with the strong enough electronegativity to strip the last component in the ETC of its electrons.
Then,